In this video, we're going to talk about cells. So we have sort of three major aspects that we're going to look at. We're going to look at the cell theory, we're going to look at the plasma membrane, and then we're going to compare prokaryotic cells and eukaryotic cells. If we look at the cell theory, there are five major components of the cell theory. So number one, all living organisms are composed of cells. The cell is the single smallest unit that can survive by itself. All cells come from pre-existing cells. Nobody really knows about the very first cell, but all the cells that exist now have to come from cells that already exist. All cells have the genetic material DNA. So specifically DNA, which can be a little bit different in viruses, and viruses are not technically considered living things. Talk about that in a second. And then all cells are either prokaryotic, which are basically our bacteria, or eukaryotic, which are the complex cells that make up us, humans, plants and animals and fungus. I want to talk about viruses. They are not considered living things because they are not composed of cells. They are made of a protein capsule. They can't survive independently. They have to live within other cells. And their genetic material can be DNA or RNA, depending on the type of virus. And viruses can't be eukaryotic or prokaryotic, but they can infect both. So in this video, we are just looking at prokaryotic and eukaryotic cell types. Cells are very small. I want to show you, see this dot here. Oops. This dot. That dot is about 1 millimeter. If you take that dot and you divide it by 1,000, oh, my dot went away, there. Then each of those 1,000 dots is 1 micrometer. So human cells, or eukaryotic cells, can be anywhere from about 5 to 20 micrometers. And then prokaryotic cells are somewhere between 2 and 10 micrometers. If we look over here at this diagram, this is showing a human egg cell, which is actually the hugest cell in the whole body. It is 100 micrometers. And then all of these little guys on top of the egg cell, those are sperm. They are about 5 micrometers, and they are the smallest human cell. 100 micrometers is about the maximum distance that, or the maximum diameter a cell can be, because we have to diffuse oxygen. Oxygen has to be able to get from the outside of the cell into the inside of the cell where we have the nucleus and the DNA, so that, you know, chemical reactions can occur and energy can be made. This is the maximum size. So what happens if an organism is going to get bigger? It just has to have more cells. The other thing is that not all cells are round. Sometimes cells have structures, so they can kind of technically be bigger if they're not round. If we look at the intestinal layer inside of our digestive tract, we have sort of structures that we call villi and microvilli, and this increases the surface area for digestion. Also, if we look at neurons, a neuron can be more than 1 meter long. So they can be very long because they are still very small in diameter. So even though this neuron could be 1 meter, the diffusion distance for oxygen is still very small because there's a lot of surface area. This diagram is showing the increased surface area of microvilli in an intestine. Next, we're going to look at our cell membrane. So all cells have a membrane, and the membrane is composed of these very special fats called phospholipids. Phospholipids have two different components. The head part is hydrophilic, so it likes water. And the tails, there are two tails, and they are hydrophobic, so they do not like water. So when these phospholipids come together, they form a bilayer because there's two rows of phospholipids and they come together naturally. If you took a handful of phospholipids and threw them into a glass of water, all of the tails would be attracted to each other and the heads would be attracted to the water. So when we put them together in a membrane, the inner portion is hydrophobic and the outer portion is hydrophilic and likes water, whether it's inside the cell, the cytoplasm, or outside the cell, the interstitial fluid. So cell membranes act like a barrier. They prevent certain molecules from just moving into the cell. And some things can move easily in and out of the cell. In another video, we're going to look at diffusion and how we transport things across the membrane. Right now, I want to just show you that the cell membrane has all kinds of molecules in it. The phospholipids make up the bulk of the plasma membrane. We have cholesterol molecules that are integrated that help give the plasma membrane stability. So the plasma membrane, because these little phospholipids are bent, they're unsaturated. So the plasma membrane is like oil. So the two main fats are phospholipids and cholesterol that make up the membrane. But we also have membrane proteins. Membrane proteins are extremely important to the functioning of the plasma membrane because proteins are what allow specific substances to come into the cell or to leave the cell. So some things can diffuse, like oxygen can always go into the cell, but other things like nutrients or hormones have to bind to proteins on the cell membrane or transport through proteins. When we look at different types of proteins that you would find in a plasma membrane, they have some very specific functions. Here's a list of specific categories of proteins that you could find within a cell membrane. First, we have receptors. Receptors act like a signaling molecule. So let's suppose you produce the hormone insulin. Insulin has to bind to a specific receptor in order to have its function of regulating blood sugar. When you think of receptors, think of things like hormones. Antigens act like cell markers. An example would be the blood types, like having type A blood or type B blood or type O. Then these antigens help to label the cell. Enzymes catalyze chemical reactions. We can have enzymes inside the cell and outside the cell. Some are in the bloodstream, and sometimes there are enzymes in the cell membrane that help to catalyze chemical reactions. Transport proteins. We're going to look at a whole video about how we move things in and out of the cell membrane, like nutrients and waste products and different substances. Adhesion. Sometimes cells need to stick together to form a tissue. So adhesion proteins are found within the cell membrane and they can help cells stick together to form a tissue. And lastly, gap junctions. Gap junctions are important because they act like channels that allow cells to communicate. So signaling molecules can move directly from one cell to another cell. Cells can either be prokaryotic cells or eukaryotic cells. Prokaryotic cells are like bacteria. all the different kinds of bacterial organisms. Some of them make us sick. Some of them are very healthy and beneficial and live in our digestive tract, help us digest food. And then we have eukaryotic cells that are the complex cells that make up things like protists, fungus, plants, and animals like my dog, and also humans. We are composed of eukaryotic cells. So we're going to look at the difference between those, and we're going to look at the structure and function of different aspects of those cells. Here's an example of a typical prokaryotic cell that has all the possible features that you might find in bacteria. Bacteria is plural. A single bacterium is a singular organism. So what do we have here? We have a plasma membrane. All cells have a plasma membrane. Bacteria can also have a cell wall. Now, this cell wall is made up of a substance called peptidoglycan. It is not cellulose like a plant cell wall. So the cell wall of bacteria is not the same as plant cell walls. Sometimes bacteria can have an extra protein layer called a capsule. This is for extra protection because bacteria are single-celled organisms. They don't have skin to protect them from environmental, pH or temperature or whatever. Inside the prokaryotic cell, we have a nucleoid region. Bacterial organisms don't have organelles inside the cell that are surrounded by a membrane. Eukaryotic cells do. So in bacteria, they don't have a nucleus. We have a nucleus in all of our cells that contains our DNA. So in bacteria, the DNA is in this nucleoid region. Also, bacteria have one single chromosome. Humans have 46 chromosomes. Different organisms have different numbers of chromosomes. So in bacteria, they typically have one chromosome. Sometimes they have a little extra piece of DNA called a plasmid. We'll talk about that in other videos as well. The fluid component of the cell is called the cytoplasm, and it includes all of the dissolved substances that are inside the cell. There are ribosomes. Ribosomes are these little dots. They are not surrounded by a membrane. Their important role is to build proteins. So remember, DNA is used as the code to produce RNA molecules that is used as the code to produce proteins. So the protein is produced on the ribosomes. And then lastly, some bacteria can have these extra structures. So some bacteria have these little pili. They kind of look like little hairs. The purpose of these is to exchange genetic material, like perhaps this little plasmid, which might have a few extra genes on it. The bacterial cell can copy it, move it through its pili, connect with another bacterial cell, and exchange that DNA. And lastly, some bacteria have a flagellum, and this helps it to swim and move around. This functions just like a sperm tail, made of microtubules. So those are the main structures of a prokaryotic organism. Not all of them are found in every single bacterial species, but those are the primary structures that you would find in prokaryotic cells. We're going to make a chart. So this chart is a great way of organizing all of the information. Charts are awesome. So we are going to look at prokaryotic structures and their function, and then we're going to look at eukaryotic structures and their function. Now we're going to look at eukaryotic organisms. So we're going to look at a single cell, and we're going to compare with prokaryotic cells and eukaryotic cells. Do they have anything in common, or are they completely different? Here's a typical eukaryotic cell. Eukaryotic cells are complex, and that means they have organelles. Organelles that have membranes. Eukaryotic cells have a plasma membrane. So this is our phospholipids and our cholesterol and our protein molecules that are going to let things move in and out of the cell. Okay, so that's the same as a prokaryotic organelle or prokaryotic cell. And then we have all these different organelles that have different functions. We're going to look at each of those functions. Eukaryotic cells have cytosol, which is the fluid, or cytoplasm, which is all of the liquid within the cell. And instead of a nucleoid region, we have a nucleus, which is where we have the DNA. So we're going to look at each individual structure and we're going to compare. So first of all, let's just have a quick look at our chart and we're going to look at the things that a prokaryote and a eukaryote, those cell types, what do they have in common? Are eukaryotic structures that are the same as a prokaryote. They have a cell membrane, they have cytoplasm, and ribosomes. Number one, we have a nucleus. Nucleus has a few characteristics, a few structures that are interesting. Okay, number one, we have chromatin. Chromatin is our DNA. Chromatin is just a word we use for the DNA when it's all loose and not condensed into tight little chromosomes. When you see kind of these X-shaped little chromosomes in diagrams, we call that a chromosome. And that is when the DNA is super coiled. We can see DNA in this structure when we have cell division and the chromosomes are going to move. How many chromosomes do humans have? We have 46 chromosomes. Inside the nucleus, we also have this sort of dark, blobby area called a nucleolus. The purpose of the nucleolus is to make some RNA molecules, specifically ribosomal RNA. Okay, I'm going to remind you again, remember DNA is our genes that is used to code for RNA. RNA is used to code for proteins. The RNA, we have different kinds of RNA. So ribosomal RNA is the kind of RNA that helps to make up the ribosome. This is produced in the nucleolus. The nucleus has a nuclear membrane, also called a nuclear envelope. It is a double layer. There are two. So here we have our normal cell membrane structure, the heads, the tails. This is a bilayer. The nuclear membrane is two of these. So it's actually folded on itself to make a double membrane. And then we have pores. These pores are important because it helps large molecules to move out of the nucleus, and a good example would be RNA. So transcription, the production of RNA, happens in the nucleus. DNA codes for RNA. That RNA then has to go to a ribosome to make protein. So that large RNA molecule leaves the nucleus through the pores. Next we have endoplasmic reticulum, and there are two kinds. We have rough and smooth. They have two major functions. So first of all, notice here that the rough endoplasmic reticulum has dots all over it. Those dots are ribosomes. What do ribosomes do? They make protein. So the main purpose of rough endoplasmic reticulum is to make protein. Smooth endoplasmic reticulum does not have ribosomes. Its main function is to make fats or carbohydrates. And do you remember the kind of carbohydrate that we can store a little bit of in our body? There's a kind of carbohydrate that we can store in our liver and our muscles. Smooth endoplasmic reticulum in the liver would make this carbohydrate. It's called glycogen. Different carbohydrates, we can sometimes put sugars together and attach them to a protein. And then we have glycoproteins. The smooth endoplasmic reticulum makes fat and carbs, and the rough endoplasmic reticulum is for making protein. Next we have the Golgi bodies, also sometimes called the Golgi apparatus or the Golgi complex. And it is a membrane structure of tubes that kind of look like this. And look at where it's located in the cell. The rough endoplasmic reticulum is located close to the nucleus. The DNA is in the nucleus. We're going to produce RNA that will go to one of these ribosomes on the rough endoplasmic reticulum, and we make protein. Sometimes those proteins need to be modified, so they will be carried from the rough ER through transport vesicles. A vesicle is any kind of a membrane surrounding substances inside. So if we produce proteins in the rough endoplasmic reticulum and they are inside this membrane, then that is a vesicle. So that will bring it to the Golgi apparatus. Now, because the membrane of the vesicle, of the endoplasmic reticulum, of the Golgi, they're all the same. All membranes are phospholipids. Phospholipids. They're all made of the same thing, so they can just blend into each other. And remember, membranes are similar to oil. So when this vesicle merges with the Golgi apparatus, now inside of here we have all kinds of enzymes that can help to do a bunch of different things, and most of the things that happen is modifying proteins. We process or package, put together proteins. So the protein, after it's produced on the ribosome, is not always 100% finished. So the Golgi finishes the proteins. The finished product can then be put into a secretory vesicle, which can be carried to the outside of the cell. Let's suppose this was a pancreas cell and we made insulin, which is a protein hormone, and that insulin is inside this vesicle. The vesicle can then be carried to the outside of the cell and insulin will be released into the bloodstream, and then it can have its function. Sometimes other things can be made in those vesicles that can actually function like an organelle. So one of the organelles is called a lysosome. So let's suppose we're in our nucleus. Nucleus has DNA. There's genes for enzymes that will digest macromolecules. Those enzymes are produced, they're proteins, so they're produced on ribosomes. They're packaged into a vesicle, moved to the Golgi modifies them, and then they're put into a vesicle. That vesicle could become its actual own organelle. Lysosomes are kind of like the digestive system of the cell. They contain enzymes and they break down macromolecules. There's another organelle that also kind of just looks like a vesicle. These aren't made by the Golgi. These contain enzymes as well, but also they produce things like hydrogen peroxide, and they play a role in detoxifying different substances. For example, the liver contains a high amount of peroxisomes, and it contains enzymes like alcohol dehydrogenase that breaks down alcohol. So think of lysosomes as digesting old organelles and large macromolecules. Our immune cells have a lot of lysosomes to break down pathogens, and peroxisomes contain enzymes and different kinds of molecules that help to detoxify substances. The liver has lots of peroxisomes. The immune system also has a lot of peroxisomes. Mitochondria. So mitochondria are going to be a whole topic of their own. they play an important role in making our energy. So we can break down any of our macromolecules, so proteins, carbohydrates, and fats, we can break those down into individual monomers or building blocks, amino acids, fatty acids, and glucose or fructose or galactose, and we can use those building blocks to make energy. Mitochondria are the organelle where we make most of our energy. So we're going to just look at a few specific structures or aspects of our mitochondria. So main role, they make energy, which is ATP, adenosine triphosphate. We will do a whole video about that. The mitochondria has an inner membrane and an outer membrane. So remember how we just talked about the nucleus is a double membrane. The membrane is folded. The mitochondria have two membranes as well, but it's folded in a different way, and we call it cristae. Inside the space here, we call that the matrix, and then in between the membranes is the intermembrane space. When we look at how we produce ATP, this intermembrane space will become very important. A super cool thing about mitochondria is that they probably used to be, billions of years ago, bacteria. So they are like prokaryotic cells. They're about the same size, they replicate the same way, and they contain their own DNA. They have a single circular DNA molecule, just like prokaryotes and their DNA. So that's kind of cool. So now we use mitochondria to produce energy. Ribosomes, we've talked about these already. These are the little dot structures that are sometimes free in the cytoplasm and can be on the rough endoplasmic reticulum. Their main function is to produce proteins, and ribosomes do not have a membrane. There is no membrane on this organelle. They are composed of two subunits, a large and a small subunit. Remember, DNA makes RNA, which makes protein. So how does the ribosome convert RNA into protein? If this is an RNA molecule, it weaves through this space between the large and the small subunit, and there are enzymes inside the large subunit that help to make peptide bonds between amino acids. This whole process is also going to be in another video, and that is called translation. And the last organelle that we are going to look at is called centrioles. These are structures that look like little cylinders, and they are made out of microtubules, which is just a whole bunch of tubulin proteins put together to make these long, hollow structures. The same microtubule proteins are used to make sperm tails and flagella. Okay, so these proteins play an important role in cell division, and they help cells to separate chromosomes. So that is also another video. So we are going to look at how cells replicate. We have to copy all of the DNA, and then we have to move that DNA into opposite ends of the cell. How that DNA moves is because of centrioles. So last thing, let's look at our chart.